Hello, Bio 201. This is a short video, the purpose of which is to help you learn how to use two bioinformatic resources, BLAST and GenBank. And so I will point out that there are instructions on using both of those on this worksheet, but I thought it might be helpful to have a short video in addition to help you to learn how to use these. So remember, there's an extra credit assignment that is posted, completely optional, of course, given that it's extra credit. And as I talked about in class, you must take your time and fill in this table. So really that is the extra credit assignment. You fill in this table. Once you've done that, you can open up the quiz and you can enter these names. There's no way that you will have time to figure all these things out if you do open up the quiz and then start searching. So again, the purpose of this is to do this first and then open the quiz and you can enter your answers and see how you do. So it's a maximum of 6 points extra credit. Okay guys, so the first thing we do is go to BLAST. And so BLAST stands for Basic Local Alignment Search Tool. And if you look up at the top of this page, you'll see the NIH little symbol there. And so this is a website that's run by the National Institutes of Health. Remember, we've talked about NIH a number of times in our class. You'll recall that Francis Collins is head of NIH and that Dr. Fauci, who you hear from frequently these days, is an NIH epidemiologist. So how do we use it? I might first point out, how do you get here? Well, I just Google BLAST and this page will come up. We go down, we could either look for nucleotides or proteins. We are looking for nucleotides, so we'll click on nucleotides. And a little blank comes up here, and so it says enter query sequence. In other words, the sequence that you are looking for. And so we could either enter the accession number, which is a number in GenBank, a resource we'll get to momentarily that's associated with the sequence, or we could enter the FASTA sequence. And so that just means the order of bases. And so what we're going to do is go over here to our worksheet. Here's the worksheet, and I'm going to go down and we'll just pull up a random sequence from the worksheet. So remember, this is a sequence of nucleotides, a gene, in other words, from an organism. I'm going to paste it into the blank there. I'm going to go down here and I'm going to modify things a little bit. I'm going to click on somewhat similar sequences, and then I'm going to click on BLAST. And so what this website is doing is comparing this sequence that I entered to every other sequence that's found in this database called GenBank, which is the largest database of nucleotide sequences on the planet. And so it will take it just a minute. So remember, it's comparing that sequence, the order of A, C, T's, and G's. Okay, so here's my result. Now I'm going to scroll down here and look at the results. And so what I see is the query coverage. In other words, how much of my sequence, how long my sequence is relative to the sequence in the database. And so in this case, it's 94% as long as the sequence in the database. And here's the percent identity. And here it's 96.36%. So that looks pretty good. In fact, I see the first three entries are all 96 to 97%. And then the third one drops down to 86. And then as I go down here, there's lots of other species here, but they're all lower than that. So I'm interested then in this particular sequence. And so I click on this sequence. I can see a nice alignment there. And, if I may know this scientific name or I may not. This is a fish, so I happen to know it. But, of course, if I studied some other group of organisms, I wouldn't. So what I'm going to do is just copy it. And I'm going to put it into Google here. And I'm going to see that it's a freshwater drum. And so I'm going to call that a victory there. So the first part of your assignment involves exactly what we just did. You are going to look at these 7 sequences shown here, and you're going to identify them. And so you're going to place the common name and the scientific name in this table. So that's it as far as that goes. Now the second part involves matching up primers to these sequences. And so once again, I have these 7 sequences, but then down here, I have 7 primers. Now, what I'll tell you is this. These 7 primers match the 7 sequences above, but only, each primer is specific to a particular sequence. And so in order to complete this, what you're going to need to do is to use a function in BLAST that allows you to compare 2 or more sequences. So all you're going to do is to go back to BLAST, which we could do here. And let me just backpedal a couple times. And so now I am here on the first page. I'm going to go up here and I'm going to click this little box, align 2 or more sequences. And when I do, look at this. Another box pops up down below. This allows me to make a comparison between 2 sequences. And so it's not comparing it against all the sequences in the database and figuring out what it is, but it's simply comparing one sequence to another. So you actually are going to use this for 2 parts of this extra credit. You're going to use it for what I just described, and you're going to use it for this final question here. So the final question is, what percent similarity exists between the cytochrome B gene and the sand roller and the Sierra Nevada mountain yellow-legged frog? And so this question allows you to do a little bit of minimal web sleuthing here. And so essentially what you're going to need to do is figure out the scientific name of both of these animals, and you're going to use that to find the sequence, cytochrome B sequence of these animals. So it's really and then you're going to compare them. So it's really pretty easy. So let's go back here to BLAST, and then let's introduce a new resource, GenBank. And so I always get to GenBank by Googling GenBank. GenBank is another resource that's managed by the NIH. And so this is the largest depository of nucleic acid sequences in the world. And it's, when students do research with me, we always enter our sequences into GenBank. It's not that big a deal these days, certainly, to do that, but it's a nice little thing you can put on your CV. And there are millions of sequences that are found here. And so it's a public database. Anyone can access this. And it's extremely useful. So people are constantly using it. Occasionally you get emails from people that are using a sequence that you have on GenBank. So how do we use it? Well, we want to search for a nucleotide sequence for sure. And then let's just say I want to find the cow cytochrome B gene. Cow cytochrome B. I search and to see what GenBank has to offer, and I look down here, and these look pretty good. So here is 1,140 base pair DNA sequence of cytochrome B. I know that that's right about the overall length of cytochrome B. This one's is 351, so that's not quite as good. It's a partial sequence. So I'm going to go with this one. And then let's talk about how we make a comparison here. So again, that's how you're going to find the cytochrome B of both the sand roller and the Sierra Nevada mountain yellow-legged frog. So I've already found human cytochrome B here. And what I'm going to do in this case is use these accession numbers. And so look, I'm going to copy this accession number, and I'm going to go back to GenBank. That's not true. I'm going to go back to BLAST, rather. And I'm going to delete what I had there. And so I'm going to use the accession number as opposed to the overall sequence because it's just going to be a lot easier to do. And so now I'm going to go over here to the cow cytochrome B. I'm going to grab that accession number. I'm going to copy it, go back to here, and I'm going to place it in there. And I'm going to assume they're not super similar, so I'm going to say compare them using the somewhat similar algorithm. So I'm going to ask it to compare them, and it's going to take just a moment to do that, and now it has my results. And so what I find is that they're about the same length, but they're about 73% identical, which is about what you'd expect. Cows and humans are not extremely closely related, and cytochrome B is quite a variable gene that's very useful in determining the taxonomy of organisms, and so there's my answer. So that's exactly what you're going to do when you complete this last question, if you choose to do so. with the sand roller and the Sierra Nevada mountain yellow-legged frog. So let me know if you have questions as you work on this extra credit assignment.